Welcome everyone. Thank you for tuning into my talk on encoding visual attributes in capsules for explainable medical diagnoses, presented here at MICCAI 2020 with my co-authors, Drs. Torigian and Ulas Bagci. This work is motivated by the immediate need for explainability in medical imaging diagnosis, evidenced by the fact that CNN-based CAD systems have largely not been adopted into routine clinical workflows, despite their performance often significantly exceeding that of human doctors. This is not limited to the healthcare domain either. A similar hesitancy is seen in many high-risk applications, including military, security, and more. Every time, this reluctance of adoption is cited back to a lack of trust, caused by the highly uninterpretable nature of so-called black box CNN models. In machine learning, there is a known trade-off between model interpretability and predicted performance. While some work has been done to make CNNs explainable, their success has been largely limited and typically comes at the cost of worsened performance. Capsule networks, on the other hand, have shown an innate ability to encode visually interpretable features without suffering from degraded performance. Capsule networks are an attractive potential solution to many shortcomings in CNNs, where a capsule network is comprised of capsules, which are defined as a group of neurons representing a feature's presence, and more importantly, the attributes of that feature, often called the feature's instantiation parameters. In practice, this only requires two simple changes from standard CNNs. First, features are now represented by vectors or matrices rather than scalars, and this allows us to store important information about the features being learned. Second, we route this information from one layer to the next via a dynamic routing algorithm, which weights both the presence and internal relationships of the lower level features with respect to each higher level combination. And with that motivation, before we move on to discuss our proposed explainable capsule network, I need to take a brief aside to distinguish between model interpretability and explainable predictions, since these terms are often used interchangeably. In the context of this work, interpretability deals with the post hoc analysis of the inner workings of a model in relation to its predictions, such as GradCam and saliency maps, or blacking out parts of the input to see how it changes the output. Explainable methods, on the other hand, explicitly provide explanations for their predictions when making them. The argument for explainable predictions over post hoc interpretations is this. Instead of a model predicting this is a picture of a cat, and a researcher trying to break down the neural activation patterns of what parts of the image are stimulating which parts of the network, what end users would really prefer is for the model to explain its predictions just as a human would. We call this human-level explainability. When asking, why is this a cat, a human would not vaguely point to regions of the input or parts of the brain. They would answer, it's a cat because it has fur and whiskers and claws. Humans explain their classifications of objects based on a taxonomy of object attributes. And if we want our models to be explainable at the human level, they should provide end users with these same kinds of explanations. And that brings us to our research question. Can we build a capsule network to model specific visually interpretable object attributes and form predictions based solely on their combination? As an application of this research, we chose to focus on lung cancer diagnosis. Lung cancer is a perfect application within medical imaging diagnosis because radiologists already explain their predictions for nodule malignancy based on a taxonomy of attributes shown here at right. To solve this problem, we proposed an explainable multitask capsule network. An object, in this case a lung nodule, is input to our 3-layer 2D capsule network to form attribute prediction capsule vectors. Each of these vectors is supervised to encode a specific visually interpretable attribute of the target object, where the dimensions of each vector capture the possible variations of that attribute over the dataset. And the magnitude of the vector represents the attribute's presence, or in our case, its score. Then, we predict the nodule's malignancy by passing these visually interpretable capsules through a linear function and apply a softmax activation to create a probability distribution over malignancy scores, while also passing them to a reconstruction branch to perform regularization. For creating these attribute capsules, unlike in CapsNet, where parents were mutually exclusive, for example, if a class prediction is the digit 5, it can't also be a 3. Our parent capsules are not mutually exclusive of each other, where a nodule can score high or low in each of the attribute categories. For this reason, we modify the dynamic routing algorithm to independently route information from children to parents through a routing sigmoid function, where the original routing softmax employed by CapsNet enforces a one-hot mapping of information from children to parents. Our proposed routing sigmoid learns a non-mutually exclusive relationship between children and parents to allow multiple children to be emphasized for each parent, while the rest of the dynamic routing procedure follows the same as in CapsNet. Typically, in lung nodule classification datasets, a minimum of 3 radiologists provide their scores on a scale of 1 to 5 for nodule malignancy. Previous studies in this area follow a strategy of averaging radiologist scores and then attempt to either regress their average or perform binary classification as above or below 3. However, such approaches throw away valuable information about the agreement or disagreement amongst experts. To better model the uncertainty inherently present in the labels due to inter-observer variation, we propose to directly predict the distribution of radiologist scores by fitting a Gaussian function to the mean and variance as the ground truth for our classification vector. This allows us to model the uncertainty present in radiologist labels and provide a meaningful confidence metric at test time to radiologists. Nodules with strong inter-observer agreement will produce a sharp peak as the ground truth during training, in which case predictions with large variance, i.e. low confidence, will be punished. Likewise, for nodules with poor inter-observer agreement, we expect our network to output a more spread distribution and will be punished for strongly predicting a single class label, even if correct. At test, this variance in the predicted distribution provides radiologists with an estimate of the model's confidence in that prediction. XCAPS, being a multitask framework, has 3 losses in its overall objective function. First, for the reconstruction branch, we choose to reconstruct only the nodule region of the input, masked by the ground truth segmentation. We then compute the mean squared error between this and the reconstruction branch output. Next, for our 6 attribute predictions, we compute the mean squared error between the network prediction and the normalized mean of the radiologist scores for each attribute. And lastly, for predicting malignancy, we compute the KL divergence between a Gaussian distribution fit to the mean and variance of radiologist scores and the softmax over the malignancy output prediction vector. The total loss is the sum of these functions. We performed experiments on the LIDC dataset where at least 3 radiologists annotated 646 benign and 503 malignant nodules, excluding nodules of mean score exactly 3. Our method was compared against the state-of-the-art explainable CNN for lung cancer diagnosis, HSCNN, which is a deep, dense, dual-path 3D CNN, as well as 2 non-explainable 3D CNNs and the original CapsNet. The results of our experiments show that supervising the attributes learned within the vectors of our capsule network significantly improved our performance over CapsNet, while a CNN-based method, which built an identical explainable hierarchy of first predicting attributes, then malignancy, suffered from degraded performance compared to its non-explainable counterparts, shown here in this symbolic plot. Here are the quantitative results of our experiments, where our simple 2D 3-layer XCAP significantly outperformed the explainable HSCNN on predicting attribute scores, while also achieving a higher malignancy prediction accuracy, with performance comparable to that of the non-explainable deep multi-crop and multi-scale 3D CNNs. We also performed an additional 3 ablation experiments, which can be found in our paper. And that'll conclude this presentation. I would like to thank the NIH for providing funding support for this project. And thank you for everyone for joining. If you'd like to view the code or any of the project pages, you can follow the links provided here.